Welcome back, everyone. This is Living Systems 1, and tonight's lecture, or the first of two lectures, is the chemistry of life. So I hope that by the time we get to this lecture, you're going to appreciate really how chemistry underpins so much of what's critical in the support of living organisms, particularly right down at the cellular level. And of course, everyone appreciates that all living organisms, whether we're talking about animals, plants, bacteria, they're all made up of cells. So let's start by considering this first point here, chemical and energy transformations in cells. So you can imagine that all cells have to be in a highly organised state. They consist of probably thousands of chemical reactions and energy transformations that are occurring at any one time. And these chemical reactions, these energy processes are absolutely critical in order for the cells to survive. Think about it in this way. The cells have to constantly be making proteins. They have to be constantly be transporting nutrients into the cell, transporting wastes out, maintaining and repairing cell components, and so on. And the sum of all these chemical reactions and energy transformations that are taking place is referred to as metabolism. The chemical reactions that occur within cells occur along these step-by-step in these step-by-step sequences. So, for example, you may have reactants reacting together in a chemical reaction to form products. And then those products will act as the reactant for the next chemical reaction. They react to form new products, and so on and so on. So you get this sequence of chemical reactions along what we call a metabolic pathway. So these sequences of chemical reactions occur along metabolic pathways. And metabolic pathways might be linear pathways, where the final end products are very different to the starting reactants, or they could be cyclic metabolic pathways. But that's something that we'll explore later on. So let's consider the term now energy. In its simplest terms, energy can be defined as the capacity to do work. There's two major types of energy: potential energy, which is stored energy, and kinetic energy. So let's think about this stored or potential energy within living organisms, within cells. And the most important one is the energy stored within covalent bonds. So everyone knows what covalent bonds are, and the electrons in those covalent bonds represents a source of energy which is utilised by cells. Kinetic energy, on the other hand, is expressed as movement, the energy of motion. So it's important to note here that whether we're talking about a molecule or an atom, and whether that molecule is a very small molecule or a very large molecule, all these different types of particles have their own intrinsic energy, their own intrinsic kinetic energy. In other words, they're not just static, immobile particles. They all move. They all move around, and that's the source of kinetic energy, that energy of motion. All right, energy transformations are governed by the laws of thermodynamics, and you've already been introduced to the first law of thermodynamics. Can anyone remember what that law was? And this was introduced in one of last week's lectures. Who remembers this? The first law of thermodynamics. If you can't remember, that's fine.
No.
No? No, can't remember. So remember last week we talked about balancing chemical equations. Mm-hmm. And we started that discussion by saying that atoms cannot be created or destroyed.
Yep.
Yep. That equally applies to all matter, and it equally applies to energy. So that's the first law of thermodynamics, that atoms or matter or energy cannot be created or destroyed. It can simply be transformed into a different form. The second law of thermodynamics, which we'll be looking at here, says that the entropy of the universe is increasing. The entropy of the universe is increasing. Any idea what's meant by that term entropy?
Is it like the reactions taking place, or it's not quite that.
Yeah, no, it's related to. It can be related to reactions, definitely. So when we're talking about entropy of the universe, and of course this is referring to the whole universe, but we can also talk about entropy within a chemical reaction, it's referring to disorder, randomness. It's the law is stating that the entire universe or a smaller system, say within your test tube in the lab, for example, will proceed towards entropy or towards disorder or randomness unless you put energy into that system. And the best way to think about this, and I like to use the example of a car. You buy a brand new car and a lot of energy has gone into that car, into the materials, to make the materials and to assemble the car. Now, what happens if you're anything like me and you just drive that car into the ground? You don't maintain it, you don't service it, it starts getting oil leaks and the bushes start to wear and it starts to rust and the whole thing falls apart because you're not maintaining it. So the car is proceeding into disorder, into a disordered state. Okay, so that's one nice way of looking at entropy or disorder. And in the same way as we're just discussing the car, all physical and chemical processes will degrade unless energy is put into the system. So if you don't keep fuel molecules going into your cells to provide energy, those cells will eventually degrade and die. They need that constant input of energy to keep them alive. All right, now let's apply that law of thermodynamics, the second law of thermodynamics, to chemical reactions. What we're seeing here is a chemical reaction where A reacts with B to form the products C plus D. But have a look at this arrow here, this double-headed arrow. Anyone know what that means, the double-headed arrow?
Is that the equilibrium?
Well, what the double-headed arrow is denoting is that A can react with B to form C and D, but C and D can react with each other to reform A plus B. So this is called a reversible reaction. Reversible reaction, it can go in both directions. All right, what would happen if you just had a single arrow pointing from left to right? What would this mean?
The reactants will end up making the products.
Yes, and the reaction stops there. Yep. So A and B will react to form C and D, and the reaction stops there. This is called the forward reaction, a forward reaction. And the reaction is said to go to completion. So this is a forward reaction. So once this takes place, the products remain as they are. They don't react to reform the starting reactants. What if we had just a reverse arrow? Yeah. So this is talking about a reverse reaction, but what it more often means is that A and B never react to form C plus D. The reaction just does not take place. And this actually can be applied very nicely to the example of photosynthesis. Photosynthesis would never proceed, if we were looking at the actual chemical equation for photosynthesis, would never proceed unless you put energy into that system. And of course, that energy is derived from sunlight. Now, what you can also see, what you might also see is this: a forward arrow and a little reverse arrow. And what this means is that most of A and B will react to form C plus D, most but not all, or that it reacts to form C plus D, but a little bit of C plus D will reform A plus B. Or you could see a small forward arrow and a large reverse arrow. So only a small amount of this will react to form this. Okay, another way of writing this reversible reaction here is to use a large forward arrow and a large reverse arrow. Okay, so that's a different way of doing it. All right, so let me just clear all that so that you can see what's going on here. All right, so let's take our reversible reaction once again. When A plus B react to form C plus D and C plus D reacts to reform A plus B, and when the forward reaction is the same as the reverse reaction, and there's no net change in the concentration of the reactants or products, then the reaction is said to be at equilibrium. Okay, so remember that when the forward and reverse reactions are equal and there's no overall change in the concentration of reactants or products, the reaction is said to be at equilibrium. All right, how does this work in living cells? It's not very useful at all. It's not useful. When a reaction is at equilibrium, it's actually not performing any useful work. It's said to be in a maximum state of entropy or disorder. For chemical reactions to be useful in living cells, they need to be out of equilibrium. So the chemical reaction has to be pushed somehow to one side. For example, energy has to be put into it to push it to one side so that it proceeds to carry out that chemical reaction. Okay, the position of that equilibrium in the chemical reaction is described by what's called the equilibrium constant. That's abbreviated to KEQ, and the equilibrium constant simply equals the concentration of the products divided by the concentration of the reactants. If the reactants and the products in that chemical reaction contain the same chemical energy per molecule, then the equilibrium constant will equal 1. And of course, as I said earlier, that is not very useful for living cells because it won't actually perform any work, any useful work. On the other hand, if the reactants and the products contain different amounts of chemical energy, and I'll illustrate this a bit later, then the equilibrium constant, KEQ, will not equal 1. The value will be less than 1 or it'll be greater than 1. In other words, the reaction will be out of equilibrium. Okay, a more useful measure of the energy potential found in a chemical reaction is represented by what's called free energy, Gibbs free energy. So it's given the symbol G. Gibbs free energy represents the maximum amount of useful work obtainable from a chemical reaction. Even more useful representation of that is what's called the change in free energy, delta G. So just note that this symbol here is referred to as delta G. This is the change in Gibbs free energy. So this is the usable portion of energy from a chemical reaction, because no chemical reaction is 100% efficiency. There's always some loss, usually as heat. All right, so what does delta G, what is that referring to, or what does it include? Delta G depends on the change in heat content. That's represented by this delta H symbol, and this is determined by the making and breaking of chemical bonds in molecules. Delta G also refers to the change in entropy, change in disorder of the system. And this is given the symbol delta S, where S is a symbol for entropy, and this is determined by the molecular organisation of the system, and then also the temperature in degrees Celsius. So this is the formula here, which you don't have to remember. You'll be happy to know. All right, that change in free energy, delta G, is related to the concentration of the reactants and the products. When the value for delta G is less than 0, that is, it's a negative number, then energy is said to be released in this type of reaction. When energy is released, the reaction is called an exergonic reaction. Normally, energy is released in the form of heat, so we often also refer to this as an exothermic reaction. And I'll just put that in the box for you. Exothermic, so exergonic or exothermic. These reactions that actually release energy are spontaneous reactions. They're the forward reactions with that single arrow pointing from left to right. These are the reactions that will just go spontaneously to completion. Energy releasing reactions. Okay, when delta G, so the change in free energy, is greater than 0, that is, it's a positive number, then energy and energy input is required to drive this reaction. And the reaction is said to be an endergonic reaction or endothermic reaction. Requires an input of heat or requires an input of energy of some type to drive the reaction. This is a non-spontaneous reaction. The reaction will not proceed unless energy is put into this. Now, living cells contain both types of reactions, both exergonic or spontaneous reactions and the endergonic or non-spontaneous reactions. And what they do is they couple or link these two types of reactions together so that the energy provided by one reaction, by the exergonic reaction, provides the energy required to drive the endergonic reaction. This is a really common theme in all cells. All right, let's now illustrate that process and also try to understand why the values of delta G might be negative values or why they might be positive values. All right, let's look at this first graph here. So this is the energy level, so from low energy to high energy, and along this axis we have the chemical reaction proceeding. Let's start with the reactants. This represents the energy level of the reactants, and let's give that level, that dotted line, a value of 0. Okay, so the reactants start with this much energy. Look at the products that will form. They are lower in energy, so we've got a difference in the energy content between the reactants and the products. All right, when this reaction proceeds, first the energy level has to increase, and then it proceeds spontaneously to a lower energy level to form the products. Let's think of this in another way. Let's say we have a hill. We go down to the bottom, and we have a ball at the top of that hill. All right, in order for that ball to roll down the hill, we need to give it a push, don't we? But once you push it, once it starts rolling, it will just proceed on its own down the hill. This is why we call this a downhill reaction, a spontaneous downhill reaction. But remember, we said we have to push the ball first to get it going. We have to input a little bit of energy, and that's what's represented here by this initial increase in energy before the spontaneous downhill reaction proceeds. This little bump in the process is referred to as the activation energy. So these reactants need to actually have enough energy initially in terms of the way they collide with each other or the speed at which they collide with each other. They have to have enough energy at that moment to break existing bonds so that they can react with each other. So this is called the activation energy. All right, now let's turn our attention to delta G. Here's our delta G. If this is zero energy, and this is the energy level of the product that's produced, you can imagine that the difference here will be a negative number. And this is why an exergonic reaction, that is an energy producing reaction, is given a negative value. That is a number less than 0. You can also start to see why energy is released, because as the reaction proceeds, it's going from a higher energy level down to a lower energy level, and that excess energy is released here. All right, let's now look at this example at the bottom, in the bottom graph. So in this example, we've got the reactants here, lower energy level. The products that will form have a higher energy level. So in order to drive that, we need an input of energy, which can come from here in living cells. So remember, we said that in living cells, these types, two different types of reactions, are coupled together or linked together. Okay, that energy that's supplied drives what we call a non-spontaneous or uphill reaction. And we can think about this once again by thinking of the ball at the bottom of the hill. And in order to get it up the hill, we have to push it all the way up. Okay, that's our input of energy. That's our uphill reaction. Why is delta G a positive number? Because we're starting at this energy level, which we can say is 0, and we end up here at a higher energy level, and therefore delta G will be a positive number. Okay, now let's consider how fast these chemical reactions proceed, the rate of reaction. The rate of reaction towards equilibrium is independent of the equilibrium constant, and it's independent of delta G. The speed of the reaction depends on the kinetic energy of the reacting molecules, that is, the energy of motion, the energy of movement of those molecules. And as we said on the previous slide, that little hump before the spontaneous reaction starts to proceed downhill is called the activation energy. This is the minimal level of energy necessary to break existing bonds in the starting reactants at the moment that the molecules collide. How is the rate of reaction increased? Very simply, it can be increased by applying heat. So if we increase the temperature, you can imagine that that increases that kinetic energy of movement of the molecules. They start moving or vibrating more rapidly, and if they do that, if they're moving faster, then they're more likely to contact with each other in the right orientation and with the required speed to provide the energy required for the chemical reaction to proceed. That is, to provide that activation energy to drive the chemical reaction. But let's think about increasing the temperature in living cells. Living cells operate within a very strict temperature range. If cells just increase their temperature, they're going to damage a lot of those internal molecules that are required to sustain life. We'll talk more about that a little bit later on. So cells can't use heat to increase the rates of reaction, but they need to do something because a lot of reactions proceed far too slowly to be of any benefit to living cells. They need to speed up the rates of these chemical reactions, and they do so by using catalysis. They use catalysts, biological catalysts, and these catalysts will reduce that activation energy of the reactants. So when we talk about catalysis, what does that mean to everyone, that term catalyze or catalysis? Any idea what that means?
Something to spur something else.
Sorry?
Something that you use to spur something else.
Yes, yeah, you could say it in that way. Yeah, to spur something on. Yes, so you can say to catalyze something is to accelerate something. Yes, to speed something up. Yep, absolutely right. Accelerating the rate of a reaction, catalysis. All right, this is just a very simple representation of what would occur in the absence of a catalyst. So this is a population of molecules here, and each molecule will have a slightly different amount of kinetic energy, its own kinetic energy. So within this population, only a small fraction of molecules actually have enough energy content to react when they collide with each other, if this is the minimum energy content needed for the reaction to occur. But in the presence of a catalyst, then that minimum energy content that's required is reduced. So now a greater proportion of those molecules can actually react. And that means that the rate of reaction increases. All right, here we are. Biological catalysts are enzymes. Enzymes are a specific group of proteins that act as biological catalysts. They act to lower the activation energy in the starting reactants, which we are now going to refer to as substrates. It means the same thing. It really depends on what area of science you're studying. Reactants, substrates, same meaning. All right, so keep in mind that there's lots of different groups of proteins that you find in cells, and enzymes represents only one group of proteins. Most enzymes are proteins. There are some exceptions to this rule. All right, so what does an enzyme do? An enzyme will bind to a substrate, in other words, to a reactant, to form an enzyme substrate complex. Within this enzyme substrate complex, this is where the chemical reaction is catalyzed. At the end of that chemical reaction, the enzyme will no longer have any affinity for the new product that's been formed. So it actually releases the newly formed product. The enzyme itself is unchanged. It retains its original form and its original function, and it can go away to find a new substrate to bind to. Now, let's look at how incredibly important enzymes are in living cells. This is a great illustration, this chemical reaction here. This is a very important reaction in animal cells, but even in plant cells also. So let's look at this. Carbon dioxide will react with water to form H2CO3, which is carbonic acid. This reaction will proceed on its own without any enzyme catalyst, but it proceeds very slowly. In the absence of the enzyme, about 200 molecules of carbonic acid will be formed per hour. 200 molecules per hour. Now consider what happens in the presence of the enzyme. Carbonic anhydrase is the enzyme. It will bind both carbon dioxide and water and catalyze this same reaction, but it does it at an incredible speed. It will enable the production of 600,000 molecules of carbonic acid per second. That's incredible. From 200 molecules per hour to 600,000 per second. That's something like 10 million times faster in the presence of the enzyme. And this is what's required, this sort of process, to sustain living cells in all living organisms. Quite amazing. Oh, and just a side note, when you see the name of a protein and it ends in ASE, A-S-E, most likely that protein is an enzyme. Most enzyme names will end in ASE. There are exceptions, but this is quite a good rule that you can apply. All right, let's now look at some properties of enzymes. Enzymes have very high specificity in terms of the substrates that they will bind. Each specific enzyme will bind only one specific substrate or a very, very small group of closely related substrates. So they have very high specificity. What does an enzyme actually look like? Let's look at this image down here. So in blue, we have this enzyme protein, and the surface of the actual enzyme will form this pocket or this groove, which is called the active site. The active site is a specialized region formed from folding of the polypeptide chains. Because you remember from last week that proteins are initially made from these long polypeptide chains of amino acids, and that those polypeptide chains will fold in a very particular way according to secondary structure and tertiary structure to form the final functional protein. Well, that is after it's actually bound with other polypeptides. So this is called the active site, and the active site is where the actual substrate is bound, and we can see the substrate being bound in here in this active site. The active site is lined by those variable R groups of different amino acids. Some of those amino acids are termed substrate binding amino acids. That is, they're responsible for the binding of the substrate, but they don't participate in the chemical reaction itself. The arrangement of these R groups in the active site determines the specificity of binding the enzyme to the substrate. And keep in mind also, and I'll just get my pen to illustrate, keep in mind also, remember when we have this long polypeptide chain, we may have an amino acid here and amino acid very far away from it, but when it folds, this amino acid might end up very close to this amino acid within the active site, okay? All right, the active site contains substrate binding amino acids, and it also contains catalytic amino acids. These are the ones that are going to catalyze the chemical reaction. Okay, let's look at models that have been proposed to explain enzyme action. The first model that was proposed well over 100 years ago described the substrate as perfectly fitting the active site on the enzyme molecule, in the active site of the enzyme molecule. So it was a perfect fit. And they referred to this as the lock and key model. So this was describing a key, the way the key fits into a lock, that's how a substrate fits into the active site of an enzyme. Since then, this model has been updated, if you like, and now people generally believe that the active site in the enzyme actually changes shape when the substrate attaches to it. This is called the induced fit model, rather than the lock and key model. And this is the one that's generally followed today. It's believed that once the substrate is bound to the active site, then the substrate is placed under strain or under tension. And when that happens, the electrons start to flow more freely. They're in a more highly energized state, and this allows it to be ready for the reaction to take place. It means that bonds, covalent bonds, for example, can be broken more readily and new bonds formed more readily. So once the substrate is in the active site of the enzyme and it's bound and under strain or tension, this is called the transition state. Once the reaction starts to proceed, this is referred to as the transition state activation. All right, let's follow that once again, but using these diagrams. So to the left, in green, we have the enzyme, and you can see the active site is shown. Here's the substrate that's come along, which is called A. In the next image, we have binding of the substrate to the active site, and now you can see that both the shape of the active site and the shape of the substrate itself changes, as the induced fit model describes. So we have transition state, once the substrate's under strain or tension, and then transition state activation when the reaction starts to proceed. You can also see here that substrate A is going to be cleaved. The next part of the diagram, you can see substrate A has been cleaved by the enzyme into the new products B and C. They no longer have any affinity for the active site of the enzyme, so now they are released. The enzyme is unchanged and can go off and bind new substrate A. Let's now discuss enzyme activity, in particular the rate of enzyme activity, because this is affected by any factor that changes the shape of the active site so that it no longer can bind its specific substrate. The most important factors that affect the rate of enzyme activity, pH, which we haven't talked about yet, and we'll come to that in a later lecture, and temperature. So you can imagine if you increase the temperature, then that polypeptide chain that's gone through all the secondary structure and the tertiary structure to fold into a particular shape will start to unfold, because the bonds and the interactions that are holding that polypeptide into that particular three-dimensional shape will be broken when we increase temperature. So when the protein starts to unfold or it starts to change shape so that the active site can no longer bind, we refer to this as denaturing the protein, or simply protein denaturation. This also explains why cells can't just increase their temperature. to speed up the rates of reaction, of chemical reactions, because one of the main effects as a result of increasing temperature is that proteins will become denatured. If proteins become denatured, then obviously enzymes can't work to catalyse thousands of chemical reactions that are required. Other proteins like structural proteins and transport proteins will denature and they can no longer do their job. So an important illustration there. The rate of enzyme activity is also affected by the concentration of the substrate and also by the concentration of the enzyme itself. So let's think about this. Let's think about the enzyme in solution, floating around within the solution, and there's barely any substrate there. So these are random processes. If it takes time for the enzyme to encounter the substrate, that means the reaction will be quite slow. Conversely, if you have lots and lots of substrate there, then the chances of the enzyme encountering those substrates so that the chemical reactions can be catalysed increases, and therefore the rate of enzyme activity increases. And the same thing applies for the enzyme. If you've got lots of enzyme around, then you'll have very quick reactions. If there's not much enzyme there, then it won't proceed as quickly. All right, the rate of enzyme activity is also affected by what are called cofactors. So many enzymes require an additional component to be able to catalyse the chemical reaction. These additional components or cofactors are most often metal ions, metal cations. Let's think about some examples. Magnesium cation is a classic one. The manganese cation, the iron cation, and so on. A lot of these cations are important cofactors which are essential for enzymes to do their work. Rate of enzyme activity can also be affected by what are called coenzymes. So there's quite a few enzymes that require additional components, which may be these small molecules that are either vitamins or derivatives of vitamins. So important coenzyme vitamins include vitamins A, vitamin C, quite a few of the B vitamins, and so on. They're also required for the activity of some enzymes. And there are different small organic molecules that act as coenzymes. Okay, this slide in particular is very important for one of the pracs that you'll be doing. So weekly prac students will be doing the enzymology prac starting week 7, I think weeks 7 and 8. Prac workshop students will be also doing enzymology 1 and 2. So a really important lecture for that particular prac. All right, so what did we say about metabolism at the very beginning of this lecture? Metabolism is the sum of all the chemical reactions and metabolic pathways that are occurring in the cell. And metabolic reactions mostly result from the action of enzymes. Enzyme activity has to be regulated to prevent over or underproduction. If the cells are producing too much of a particular enzyme, more than what's required, it's wasting precious energy. If it's not producing enough of a particular enzyme, then a particular reaction will proceed too slowly, and that causes problems for the cell. So how do living cells regulate enzyme activity? There are short-term control methods and long-term control methods. Short-term control methods may include controlling enzyme activity by chemically modifying the structure of this enzyme. The most common chemical modification of an enzyme includes the covalent modification of the enzyme by adding a phosphate group, and this is called phosphorylation. covalent addition of a phosphate group. This can increase or decrease the activity of an enzyme, depending on that particular enzyme. Another short-term control method involves what's called allosteric inhibition or activation. And this involves the binding of an organic molecule to a site on the enzyme other than the active site, hence this term allo. The binding of a molecule to another site on the enzyme, the binding of that other molecule will change the shape of the enzyme. It alters the shape of the enzyme, which includes altering the shape of the active site so that it either no longer binds the substrate or it might actually activate the enzyme. It might change the shape so that the active site can now bind the substrate. Hence, we call it allosteric inhibition if it stops the enzyme from binding, or activation if this type of allosteric binding activates and allows the enzyme to bind its substrate. All right, long-term control methods of regulating enzyme activity. The concentration of the enzyme can be increased simply by making more of the enzyme. So this relates back to the actual gene within the DNA that's responsible for encoding that particular enzyme protein. That gene itself can be regulated so it's producing more of the protein, or it can be repressed so it produces less of the protein. And that's another complex process which we'll talk about later on in semester. Concentration of the enzyme can also be decreased by specifically targeting that enzyme for breakdown. So proteins are constantly being broken down, manufactured, and recycled after breaking down. The cell can actually increase the rate at which it breaks down specific enzyme proteins. All right, now we're going to talk about an incredibly important molecule. This molecule represents the energy currency of the cell. So you imagine when you ingest fuel molecules, so you had your lunch or you've had your dinner, and those molecules contain those covalent bonds, those energy-rich covalent bonds. Energy has to be extracted from them, but it can't be all used at once. It's too much energy for a tiny cell to use all in one go. So instead, the energy is extracted from the bonds of your food molecules and transferred to energy in the form of this molecule called ATP. And then ATP can provide these tiny packets of energy as required to drive cellular processes. So it's a more manageable, usable form of energy for the cell. So energy from chemical reactions, from fuel molecules are conserved in this molecule called ATP. ATP stands for adenosine triphosphate. Let's look at a molecule of adenosine triphosphate at the bottom here. Now, I wonder if anyone thinks that this looks familiar. It looks very, very similar to a molecule that we looked at last week, in fact. One of the building blocks of one of our large macromolecules looks very similar to this.
Reminds me of DNA.
Yes, absolutely. So remember last week we talked about the basic building blocks of our nucleic acids, DNA and RNA. And those basic building blocks are called nucleotides. This is almost, what we're looking at here is almost identical. So a nucleotide will have this nitrogenous base. In this case, it's adenine. Nucleotide will have this ribose sugar. Nucleotides that are found in RNA will have a ribose sugar. And nucleotides also contain one phosphate group. So the difference here is that ATP contains 3 phosphates, hence the word triphosphate in adenosine triphosphate. So these are the components of ATP. But what's important about ATP is that energy is released when this bond is cleaved. In other words, one phosphate group is released, then energy is released from here, which can drive cell processes, cell reactions. When that happens, adenosine triphosphate becomes,
Biphosphate.
sorry?
Biphosphate?
Diphosphate.
Oh, di. Okay,
Di. Yep. ATP will become adenosine diphosphate. Sorry, that should be a D. And then what can happen is that the second phosphate can be cleaved. Another phosphate can be cleaved. That releases energy as well. And this doesn't happen as frequently. And now we have adenosine monophosphate, AMP. Okay, so this is how energy is provided by ATP in the cell. This is not the only energy providing molecule in the cell, but it's certainly the most commonly studied molecule. Most is known about ATP. Okay, so that's ATP, and we'll certainly talk a lot more about ATP through the year. Okay, now we're going to look at how this relates to enzymes. ATP is synthesized through a series of complex metabolic pathways which act to extract the energy from your food molecules, which we call fuel molecules, and eventually transfer that energy into the form of ATP. One part of these complex metabolic pathways involves what's referred to as an electron transport chain or electron transport pathway. This involves a series of proteins and enzymes that are basically grouped together into a complex, and these are found on certain membranes within the cell, but don't worry about that for now. These complexes, these protein enzyme complexes, operate in a sequence with each other. And they operate in such a way so that electrons are transferred from one complex to the next complex, and then from that complex to the next complex. The complex that loses an electron is said to be oxidized, and the complex that gains the electron is said to be reduced. Who remembers redox?
Mm-hmm. Yep.
Yes, everyone remember redox?
Yep.
Here it is in action. If you don't remember reduction and oxidation, try to remember oil rig. Oxidation is loss of an electron, reduction is gain of an electron. So this transfer of electrons from one complex to the next, of course, is referred to as redox or oxidation reduction reactions. All right, this very complicated looking diagram here represents an electron transport pathway or electron transport chain. It's, as I said, a little bit complex, so I'm going to draw a very simplified version of this. But before we do, let's just look at these 2 electrons here, which will be passed from one protein enzyme complex to the next through this series of redox reactions from high energy in a stepwise fashion down the electron transport chain to a lower energy level. All right, let me just bring up my whiteboard. There we go. All right, let's see if we can draw this in a simpler way. This represents one protein complex, which we'll call cytochrome B. Cytochrome B. Okay, so this is a collection of several proteins and an enzyme, because enzymes are required to catalyze this entire process. Here we have another protein enzyme complex, a different cytochrome, and another one here. All right, within each enzyme complex, we have Fe3+, Fe3+, oh, that should read Fe3+, and Fe3+, once again, and up here, we have what's called a biological electron carrier. Don't worry about what that is or where it comes from at the moment. All you need to know is that that biological electron carrier is going to donate an electron to this Fe3+. If Fe3+, ion 3, picks up an electron, is it reduced? Will it be reduced or oxidized?
It will be reduced.
It will be reduced, yes. So it will be reduced, correct, because it's gaining an electron and it's reduced to Fe2+. Okay, everyone happy with that so far?
Yep.
Yes? All right, then what happens, Fe2+ will donate that electron to the next cytochrome complex. Here it is. So what happens to this Fe3+? Exactly the same thing. It is reduced to Fe2+. But now let's go back for a minute. Fe2+ gave up its electron to a lower energy cytochrome protein enzyme complex. So Fe2+ is said to be oxidized or reduced because it's lost an electron. Loss of electron?
It's oxidized.
Correct, it's oxidized. So it is oxidized, so it returns to this state here, to the Fe3+. Okay, down to this cytochrome complex, this Fe2+ will also give up that electron to the next cytochrome complex, a lower energy complex, so it too is oxidized back to Fe3+. Okay, same process here, Fe2+, and now what happens is that we need a final electron acceptor. Because if we can't get rid of this electron here, Fe2+ cannot be oxidized back to Fe3+. Any idea what this final electron acceptor is? And this final electron acceptor will explain why we have to breathe in oxygen.
Is it oxygen?
It is oxygen. I just gave it away, didn't I? Okay, this is why we need a constant source of oxygen, because oxygen is the final electron acceptor. It will be coupled with, it will accept that electron, or rather two electrons, and two protons to form, what do you think it will form?
Water.
Water, absolutely right. Okay, so this explains why, if we don't have oxygen, how long do you think we'll survive? A few minutes, maybe?
Yeah.
5 minutes, 6 minutes, something like that, 7 minutes. Some people go a bit further than that. because this oxygen has to be replenished all the time in cells. If it doesn't, then this process stalls completely. It
So is
completely
that,
stalls. Yep.
Is that hemoglobin? Is it?
Sorry?
Is that hemoglobin?
No, this is not describing it. No, hemoglobin is simply a carrier for the oxygen. It will carry oxygen to all the cells in the body.
Yep.
That's just a transport mechanism, if you like.
Okay.
But this electron transport chain, and we haven't actually gotten to the reason that this occurs, this movement of electrons along this, what we call a redox potential, from high energy to low energy, this movement, and remember we call movement of molecules or atoms, we refer to that as kinetic energy. Yeah, this movement actually provides the energy required to eventually synthesize, because there's a few more steps that we're not going into, to synthesize ATP. And if the cells can't synthesize ATP, they don't have energy to drive any reactions, and the cells will die very quickly. Yeah. Okay, so I think a very nice illustration of how important enzymes are, and also answers the question, why do we need oxygen? All right, so let's go back to this slide here. Okay, so hopefully that's makes it a little bit easier to understand what's going on here, the electron transport chain. This is only one part of the whole process. In Living Systems 2, we will definitely be going into all the detail of the entire process, starting with one of the molecules from the food that we ingest. And we'll describe all those metabolic pathways that are involved. All right, so we just saw a series of redox reactions. And in these redox reactions, the tendency of a particular cytochrome complex to donate or accept electrons can be measured as what's called the redox potential. And this is the symbol for redox potential. The redox reaction is said to be favourable if the electrons are transferred from a complex with a more negative potential to one with a less negative potential. And we'll look at some values on the next slide. So here we have on the right some examples of redox potentials as measured in millivolts. There's the symbol once again for redox potential. So let's look at some examples here. Here was our highest energy protein enzyme complex called cytochrome B. And this has a redox potential of plus 120. The next one down was cytochrome C, and it has a higher value, plus 220 as its redox potential. In other words, cytochrome C with the higher value has a greater tendency to accept electrons from this one here, and it has a greater tendency to donate its electrons to the next higher value one. So that's essentially how it works. Cytochrome A has a higher redox potential again, and then we look at the redox potential of oxygen. So you can see, according to this redox potential value here, oxygen has an enormous potential to receive electrons, and hence why it acts as the terminal or final electron acceptor in the electron transport chain. All right, let's finish this lecture by just briefly considering energy in fuel molecules. And everyone's familiar with the fact that carbohydrates provide energy. Lipids and proteins can also, well, lipids do provide energy, and proteins can. They're not generally used to provide energy. They have other important functions, but they can be used to provide cells with energy. So we refer to these molecules as fuel molecules. Energy can only be extracted from certain covalent bonds. The most important ones are carbon-carbon and carbon-hydrogen bonds. And we'll actually tackle these in energy extraction, in living systems too. But also carbon-nitrogen bonds have electrons that can be extracted. And we finish with a look at lipids, which of course have more energy per carbon atom than do carbohydrates or proteins. And this is because they have so many of those energy-rich carbon hydrogen covalent bonds. If you remember back to the chemical structure of a lipid, so just think about that fatty acid chain that had something like 18 or so carbons bonded together with all their hydrogens attached. A lot more carbon hydrogen bonds that you would find in, say, a molecule of glucose or in an amino acid, for example. All right, so let's summarise. We have now looked at 2 laws of thermodynamics, and these are the only 2 laws of thermodynamics that are actually relevant to biological systems. First one, energy or matter cannot be created or destroyed, but it can be transformed from one form to another. Okay, the law that we looked at today, the entropy or disorder of the universe is increasing, and we related this to chemical reactions. Chemical reactions that are not at equilibrium have the capacity to do work. Enzymes are the catalysts of life, biological catalysts found in living cells, and they act to lower the activation energy of reactions, to speed up, to greatly accelerate the rate of reaction. And metabolism, we know, is the sum of all those chemical reactions and energy transformations that are occurring in cells. So that completes this lecture.
Okay.
All right, I think we will have just a quick 5-minute break, if that's okay. And I'll be back shortly.
Okay.
Okay.
Chi?
Okay, are we all ready to go? Oh, Anthony, you're ready?
Yeah.
Yes. Yeah, I'll just make sure the others are back.
Yep.
Yep.
Oh, you're back, Liz? Yeah, Brad's ready. And just waiting for Joe. Okay, Joe, yes, he's back. Terrific. Oh, how did everyone go with the choose? Did everyone get that in on time?
Yeah.
Yes?
Yep.
Everyone else?
Yes.
Good. Okay, terrific. All right, let's proceed with our second lecture for tonight, Lecture 8. And you'll remember, or you might remember, I know it was a long time ago, week 1, we just very briefly introduced the 3 states of matter, gases, liquids and solids. And we're going to extend our studies on the 3 states of matter. So we'll start with this nice table that summarises the properties. So first property, shape and volume. Gas will expand to fill a container it's placed in. A liquid will occupy a fixed volume that takes the shape of the container. And solid has a definite shape and volume. Arrangement of particles. Gas particles are randomly arranged, disorganised and far apart from each other. Liquid particles are randomly arranged but close together. Solids have a fixed arrangement of close particles. Particle movement of gases is very fast, moderate for liquid particles and slow for solid particles. Interaction between particles. This is something that we'll discuss in more detail tonight. There are no interactions between gas particles. There are strong interactions between liquid particles and very strong between solid particles. All right, whether a substance exists as a gas, liquid or solid depends mostly on the balance between the kinetic energy of its particles, so the energy of movement of the particles, and the strength of the interactions between the particles. Now, of course, with a gas, there are no interactions between the particles and they have lots of kinetic energy. Liquid particles, there are strong interactions between these particles that actually hold them in that liquid state. And then here we have an example of a solid. This is silicon dioxide or sand. And we can see that the interactions here consist of covalent bonds between the oxygen atoms in red and the silicon atoms also. So very strong interactions there. Let's start by looking at the properties of gases, and mostly properties of gases can be summarised by what's referred to as the kinetic molecular theory of gases, which states that a gas consists of particles that move randomly and rapidly. The size of the gas particles themselves is quite small compared to the space in between those gas particles. Gas particles exert no attractive forces on each other. The kinetic energy or energy of motion of gas particles increases when you increase temperature. And when gas particles collide with each other, they rebound and travel in new directions. So you can imagine when these rapidly moving gas particles collide with the walls of a container that they're enclosed in, they will exert a pressure. And pressure is simply defined as the force exerted per unit area. Let's look at the units of pressure. The atmosphere is abbreviated to ATM, and this one we'll use quite frequently. 1 atmosphere is equal to 760 millimetres of mercury. So this is another unit of pressure, and just note Hg is the chemical symbol or the element symbol for mercury, and we'll be using this unit of pressure. 1 atmosphere is equal to 760 torr. We won't be using that one. 14.7 psi, which is pounds per square inch. You'll be familiar with that when you put air into your car tyres. And 1 atmosphere is equal to 1,01325 pascals. And this is a unit of pressure that we commonly use in plant physiology, or more commonly kilopascals rather than pascals. All right, we're going to look at what are called the gas laws. And these gas laws will describe properties of gases regardless of the identity of the gas. The first of the gas laws is Boyle's law, which relates pressure and volume of a gas. Boyle's law states that for a fixed amount of gas, the pressure and volume of the gas are inversely related. So that means if one quantity increases, the other will decrease. This is illustrated here. Boyle's law, if the volume of a gas is halved, then the pressure will double. Let's look at that here. Here we've got a cylinder of gas, doesn't matter what the gas is, and here we have a plunger. The volume is 2 litres. The pressure is 1 atmosphere. If we decrease that volume from 2 litres down to 1 litre by pushing down on the plunger, then the pressure will increase from 1 to 2 atmospheres. Boyle's law can be used to describe how we breathe. So quite interesting, when we inhale, what actually happens and how do we use Boyle's law to describe this? Okay, the rib cage, which is a muscular structure, will expand. The diaphragm moves down. This results in an increase in the volume of the lungs. Increasing the volume will cause the pressure within the lungs to decrease. So there's Boyle's law at work. Increasing the volume, the pressure will decrease. When the pressure decreases inside the lungs relative to the external air, then air will be drawn into the lungs to equalise that pressure. So think about that. We've got low pressure in here, high pressure out here, say at about 1 atmosphere. That pressure will equalise. It will move towards an equilibrium state. And to do that, air will move into the lungs. Let's look at the opposite. What happens when we breathe out? We exhale. The rib cage will contract and the diaphragm, the muscular diaphragm, is raised. And what this achieves is a decrease in the volume of the lungs. If we decrease volume, then the pressure in the lungs increases. That means we've got high pressure here relative to the external or atmospheric pressure. What happens? Air will move from high pressure out to low pressure, and the pressure is equilibrated as a result. So a nice application of Boyle's law. Our next gas law is Charles's law, which relates volume and temperature of the gas. So for a fixed amount of gas at constant pressure, the volume is proportional to its temperature in Kelvin. In other words, if one quantity increases, the other increases as well. Charles's law is illustrated here. If the temperature of the cylinder is doubled, then the volume of the gas will also double. Here's our temperature, 200 Kelvin. If we increase that to 400 Kelvin, then the volume will also increase from 1 litre to 2 litres. Okay, our third gas law is Gay-Lussac's law, which relates pressure and temperature. For a fixed amount of gas at a constant volume, the pressure is proportional to its temperature. If one increases, the other increases as well. Why do you think that might be? Why is it that we increase temperature of a gas, then the pressure increases? So imagine the gas within a container, and if you increase the temperature, why would the pressure increase?
They start moving around faster.
Correct. Absolutely right. So if you apply temperature, then the kinetic energy increases. They move around more rapidly and they exert a greater pressure on the walls of the container. So increasing the temperature increases the kinetic energy of the gas particles, and this, of course, causes the pressure exerted by the particles to increase. All right, those three gas laws so far can be combined into one equation, as you can see here. But we're going to skip this. This equation is just here for your interest, and they're summarised here. Now we're going to look at the next law called Avogadro's law, which relates volume and number of moles. Avogadro's law states that the volume of a gas will be proportional to the number of moles of that gas that are present. If one increases, the other will increase as well. Let's illustrate that. Number of moles of gas here equals 1. If we increase the number of moles to 2, then the volume will increase from 1 litre to 2 litres. So that's quite straightforward. Often amounts of gas are compared at a set of standard conditions of temperature and pressure, which we abbreviate to STP. Standard conditions of temperature and pressure. When you see this in some of your tutorial questions, remember that this is giving you the conditions already, or it's already giving you the value for temperature and pressure. So try to keep this one in mind. These are the conditions of STP: 1 atmosphere or 760 millimetres of mercury for pressure. And I'll explain in a minute why you might have to use one or the other, either atmospheres or millimetres of mercury. Standard conditions of temperature equals 273 Kelvin. At standard conditions of temperature and pressure, 1 mole of any gas has a volume of 22.4 litres. This is referred to as the standard molar volume. So regardless of the identity of the gas. This is illustrated here. So we're still on Avogadro's law. In this balloon, we have 1 mole of nitrogen, molecular nitrogen, N2. In this balloon, we have 1 mole of helium gas. 1 mole of nitrogen, molecular nitrogen, will occupy the standard molar volume of 22.4 litres. 1 mole of helium will occupy the same volume, 22.4 litres. But even though they're occupying the same volume, the actual weight or mass is different. So you might remember that 1 mole of helium equals its atomic mass or atomic weight, and according to the periodic table, that atomic mass is 2 grams. Similarly with nitrogen, if we're looking at 1 mole of nitrogen, that will equal the atomic weight or atomic mass of nitrogen according to the periodic table, which is 14, but we have 2 nitrogen atoms here in molecular nitrogen, so the total mass or weight is 28 grams. Okay, here we have an example of how to convert moles of gas to volume at conditions of standard temperature and pressure. This is here just for your interest. And let's move on to slide 23, the ideal gas law. This law, this equation rather, can be used to solve all the problems in your tutorial questions. All 4 properties of gases, that is pressure, volume, and number of moles and temperature, can be combined into this single equation, which we call the ideal gas law, PV equals nRT. Let's look at what these all represent. P is pressure, V is volume, volume must be in litres. Little n, you've already been introduced to, this is amount or number of moles. R is the universal gas constant, and T is temperature. Temperature must be in Kelvin. So let's look at R. R is the universal gas constant, and the value for R will be 0.0821 if the units of pressure are in atmospheres. So if pressure is given in ATM, then you will select this value for the universal gas constant. If the value for pressure is given using these units, millimetres of mercury, then you need to select this value for R, 62.4. All right, let's look at how to carry out calculations using PV equals nRT. Question, how many moles of gas are contained in a typical human breath that takes in 0.5 litres of air at 1 atmosphere of pressure and 37 degrees Celsius? Let's identify the known quantities and the desired quantity. Pressure, we're told, is equal to 1 atmosphere. Volume is 0.5 litres. Temperature is 37 degrees Celsius. And you're being asked to solve for the number of moles. Step 2, convert all values to proper units and then choose the value of R that contains the units of pressure. Temperature was given in degrees Celsius, but remember we said it has to be in Kelvin. This takes us back to week 1, our week 1 lecture. To convert temperature from degrees Celsius to Kelvin, we simply add 273. So we were given the value 37 degrees Celsius, simply add that to 273, and that gives us 310 Kelvin. Now, the value for pressure was given in ATM, atmospheres. So that means we have to select this value for R, 0.0821. We know this is the right one because we can see here in the units, it contains ATM, not millimetres of mercury. Right, you don't need to memorise these R values. They will be provided for you in the final exam. But you certainly do know, have to know how to select the appropriate value for R and how to incorporate that into solving the equation. All right, now we write out the equation and rearrange it so we can isolate the desired quantity on one side. All right, here's our equation, PV equals nRT. We want to solve for N, number of moles. So we need to get rid of the RT. And let's just recap what we do. So PV equals nRT. There's a lot of background noise. Can everyone hear that? Oh, what was that?
Is that better?
Is that you, Anthony?
Oh, I had the heater on.
Ah, that's a very noisy heater. Yeah, that's much better. Thank you.
That's all right.
All right, so let's rearrange this. So we need to solve for number of moles. Leave that where it is. Take these two and put them under the other side. Okay. So remember, we use this method for all the formulas that we're learning this semester. And then we can just write that out. Number of moles equals P times V, and then divide that by R times T. So here it is. Number of moles equals pressure, 1 atmosphere. Volume, 0.5 litres. R, 0.0821, and temperature 310 Kelvin. Then remember to start cancelling any like units. So we can cancel atmosphere with atmosphere here, cancel litres with litres, we can cancel Kelvin with Kelvin, and what does that leave us with? Mole Go back to what you're trying to solve for, mole, and that way you know you're on the right track. Then just calculate the answer, 0.02 mole. All right, everyone happy with that?
Mm-hmm. Yep.
Yep. Okay. All right, now we're going to look at those attractive forces that exist between molecules, and we call these attractive forces intermolecular forces. There are a few different types of intermolecular forces. We're only going to visit these three here in order of increasing strength. These are London dispersion forces, otherwise known as van der Waals forces, dipole-dipole interactions, and hydrogen bonding. Hydrogen bonds are the strongest of the three. The strength of these intermolecular forces determines whether a compound has a high or low melting point and boiling point, and thus determines whether a particular substance is a solid, liquid, or gas at a given temperature. Let's start with the first intermolecular force, London dispersion forces. These are very weak interactions due to momentary or transient changes in electron density in a molecule. The change in electron density creates a temporary dipole, not a permanent dipole, a temporary dipole. And the weak interactions between these temporary dipoles are what we refer to as London dispersion forces. All covalent compounds exhibit this type of intermolecular force. The larger the molecule, then the larger the attractive force and the stronger those intermolecular forces will be. Let's look at London dispersion forces. Here we have a molecule of methane, CH4, and another molecule of methane here. Now, at any one time, the electrons may be more dense on one side of the molecule than on the other. Remember, this is very temporary, very transient, but what this does is confers a partial negative charge, delta negative to one side, and of course this is highly exaggerated the way that it's illustrated here, which will actually confer a partial positive charge denoted as delta positive to the other side of the molecule. Same thing happens to this molecule of methane, and you can imagine there would be then an interaction between the delta negative here and the delta positive partial charges there. And that interaction in between the molecules is referred to as London dispersion forces. Our next intermolecular force is the dipole-dipole interaction. It's almost the same, except that dipole-dipole interactions are the attractive forces between permanent dipoles of two molecules rather than temporary dipoles. An example here shows a molecule of formaldehyde. And you might remember from a previous lecture that we talked about electronegativity. So remember, does anyone remember, if we compare carbon and oxygen, which of these two atoms would have a higher electronegativity value? That is, which atom exerts a greater pull on the electrons within the covalent bonds shown there?
Is it the oxygen?
Oxygen, absolutely. Oxygen has a higher electronegativity value, so exerts a greater pull on the electrons here, and this confers to the oxygen a partial negative charge, which we call delta negative, and of course the carbon, as a result, will be given a delta positive, a partial positive charge. So this is a permanent state, this dipole. This is a permanent dipole. Here's a neighbouring formaldehyde molecule, and of course this interaction between the delta negative and delta positive of these adjacent molecules constitutes what's called a dipole-dipole interaction. And of course this is a stronger interaction than the London dispersion forces. Okay, our third intermolecular force is the all-important hydrogen bonding. Hydrogen bonding is almost identical to the dipole-dipole interaction, but it occurs specifically when a hydrogen atom that is bonded to oxygen, nitrogen, or fluorine, that is these highly electronegative atoms, is attracted to an oxygen, nitrogen, or fluorine atom in another molecule. Here's a classic example, a molecule of water, H2O, and you can imagine that the oxygen is highly electronegative compared to the hydrogens. So what does that do? That imparts the delta negative charge to the oxygen, delta positive charges to the hydrogens. The same thing applies to this neighbouring water molecule, and the interaction between the hydrogen and the oxygen of another molecule constitutes what's called a hydrogen bond. Hydrogen bonds are the strongest of the three types of intermolecular forces. A very important type of bond that is found in lots of different biological molecules. Here's just one example, DNA. You might recall from last week that we described DNA as being composed of two chains or two strands of nucleotides, and these are joined together or bound together as a result of the presence of hydrogen bonds, which you can see illustrated here by these red dotted lines. Intermolecular forces are summarised here. London dispersion forces that we've just looked at are the weakest of the three. Dipole-dipole interactions, moderate strength. Hydrogen bonding are strong compared or relative to the others. All right, now let's look at how that relates to boiling point and melting point. So the boiling point is simply the temperature at which a liquid is converted to the gas phase, and melting point is simply the temperature at which a solid is converted to the liquid phase. The stronger the intermolecular forces that exist between molecules, then the higher the boiling point and melting point. Let's look at an example of this. Here we have a molecule of methane. We looked at this example, so you know that methane molecules are attracted to other methane molecules only as a result of London dispersion forces. Water molecules, on the other hand, are attracted to other water molecules as a result of the stronger hydrogen bonds. As a result, the boiling point of methane is minus 162 degrees Celsius. Compare that to the boiling point of water, 100 degrees Celsius. Melting point of water, 0. Melting point of methane, minus 183. So the stronger forces, intermolecular forces between water molecules, means that water has a much higher boiling point and a much higher melting point. And this also explains why at room temperature, water is liquid, but methane is a gas.
Silvana, does the type of intermolecular bond have anything to do with the range between the melting point and the boiling point as well?
The range, that's a good question, and I'm not too sure about that. Okay, let's look at two very similar molecules. So here we have propane and the slightly larger butane. So they both have London dispersion forces, but in this case, the larger molecule, because it has a larger surface area, will exhibit stronger attractive forces with other molecules. And we can see that illustrated here. Propane has a boiling point of minus 42 degrees Celsius, but butane has a higher boiling point of minus 0.5 degrees Celsius. All right, now let's look at the liquid state. And when it comes to describing properties of liquids and solids, we can't just apply a set of laws as we did to the gases using the gas laws. And this is because liquid molecules and molecules within a solid exhibit those different types of intermolecular forces. All right, so let's consider the term evaporation, which everyone's familiar with. Evaporation is simply the conversion of liquids into the gas phase. Evaporation is said to be an endothermic process. That is, it absorbs heat from its surroundings. The liquid will absorb heat, and then the liquid molecules can make a transition into the gas phase. So why do you think that this is an endothermic process? Why does it have to absorb heat from its surroundings in order for that transition to take place? Anyone sure about that? Have any idea about that?
Because you need to heat, the energy will convert the water molecules into.
Because
Well, the water molecule is unchanged.
Yep. So,
Yeah.
well, it goes from a solid, a liquid state to
Gas.
a gas state. So
yes.
and gases move faster, so they need more energy.
You're sort of close. You're both close. What is the heat actually doing to the water molecules to enable them to escape from the liquid state into the gas state, if you like?
More kinetic energy.
Yes, it makes them move faster, which enables them to do what?
Break down.
Break, that's right. It enables, because they're moving faster, they have enough energy to break free from the intermolecular forces. And when we're talking about water, that means that the water molecules are breaking free from the hydrogen bonds which are holding them together. This is what we refer to as the cohesive property of water molecules, that cohesive nature which allows them to stick together, if you like, is due to hydrogen bonds. When you apply heat, they move much faster and they can break free from those hydrogen bonds and escape into the gas phase. But the water molecules themselves remain unchanged as such. So that's why we say evaporation is endothermic. It absorbs heat. And it makes sense. If you consider a puddle of water outside, when the temperature is higher outside, it evaporates faster, doesn't it? So that's, I think, an easy way of looking at it. All right, the opposite reaction here, or the opposite the opposite reaction is condensation. Condensation is the conversion of gases back into the liquid phase. Condensation is exothermic, so it gives off heat to the surroundings. And we'll come back to these states in just a minute. All right, now let's consider what's referred to as vapour pressure, and how vapour pressure determines how a liquid, such as water, gets to the state where it's boiling. All right, here's our liquid water here. And at any one time, some of these surface molecules will actually have enough energy to escape from the liquid phase into the gas phase. Here they are. And of course, you know that gas molecules don't have any intermolecular forces in between them. You also know that they move around randomly and rapidly. As a result, these molecules will bounce off the surface of the liquid water and creates a pressure. And this pressure is called vapour pressure, the pressure exerted by those gas molecules. If we increase the temperature, then those gas molecules will move even faster and they'll exert an even greater pressure. When that pressure comes into equilibrium with atmospheric pressure, which is 1 atm at sea level, then that exerts enough energy into the liquid water so that all these water molecules have enough energy to start escaping into the gas phase. In other words, the water starts boiling. All right, so what would happen, given that we're talking about 1 atmosphere of pressure at sea level, what would happen if we were at the top of a tall mountain where atmospheric pressure is lower? What does that mean? What would happen if the atmospheric pressure is lower?
In terms of the temperature required to boil?
Yes, in terms of the temperature required. Yeah, if the atmospheric pressure is lower than 1 atm,
you would need less.
That's right. Then you would need less of an increase in temperature for the vapour pressure to come into equilibrium with atmospheric pressure, and therefore the water would boil at a lower temperature. Instead of boiling at 100 degrees Celsius, it might boil at 96 degrees Celsius, for example. So your cup of tea wouldn't be quite as hot. Okay, let's relate that to intermolecular forces. The stronger the intermolecular forces in a given liquid, then the lower the vapour pressure will be at a given temperature. Let's consider water, liquid water. These exhibit stronger intermolecular forces, hydrogen bonds, which keep the water molecules, or most of them, in the liquid state. That means fewer water molecules can escape into the gas phase. That relates to lower vapour pressure. If we consider methane, CH4, these only exhibit London dispersion forces with their neighbouring methane molecules. Weaker forces allow more of these methane molecules to escape into the gas phase, and more gas molecules means higher vapour pressure. So you can just see here the relationship between intermolecular forces and vapour pressure. All right, just another couple of properties of the liquid state. Viscosity is simply a measure of a fluid's resistance to flow freely. So a viscous liquid will feel thick. Compounds with strong intermolecular forces tend to be more viscous than compounds with weaker forces. So for example, water versus petrol. Not a very good example, but if you did look up the viscosity values for water and petrol, you would find that water is more viscous because of those stronger intermolecular forces, that is the hydrogen bonds between water molecules, and they are not present between petrol molecules. Okay, substances composed of large molecules such as carbohydrates, for example, tend also to be more viscous too, because large molecules don't slide past each other as freely. And this is a good example, oil versus water. And our last property of the liquid state, surface tension, which is a measure of the resistance of a liquid to spread out. So let's imagine here that we have water molecules, and if we consider just this water molecule in the centre, you can imagine that this molecule is surrounded by intermolecular forces on all sides. Whereas surface water molecules experience these same forces, but only from the sides and from below. The stronger the intermolecular forces, then the stronger those surface molecules will be pulled down towards the interior of a liquid, and therefore the higher the surface tension will be. Water has a very high surface tension because of those strong hydrogen bonds. Now let's visit the solid state. Solids can be classified as either crystalline or amorphous. A crystalline solid has a very regular arrangement of particles, that is atoms, molecules or ions, with a repeating structure. There's 4 types of crystalline solids: ionic, molecular, network, and metallic. An amorphous solid, on the other hand, has no regular arrangement of its particles. Let's look at that quickly. Crystalline solids. An ionic solid is composed of oppositely charged ions, such as sodium and chloride ions. A molecular solid is composed of individual molecules arranged in a regular fashion. So, for example, water. Not liquid water, of course, but ice. A network solid is composed of a number of atoms that are covalently bonded together. And here's our example again of sand, silicon dioxide. And finally, a metallic solid is a lattice of metal cations. And here we have pure copper. And our second classification of solids, the amorphous solids, which lack that regular arrangement of particles. And these are formed when liquids cool too quickly for regular crystal formation to occur. That is that orderly arrangement of the atoms or particles within the solid. Very large covalent molecules tend to form amorphous solids. And some examples of amorphous solids include rubber, glass, and plastic. All right, finally, a look at some energy and phase changes. And we've already touched on a couple of these. So let's take solid water or ice and converting that to liquid water. Of course, we refer to this as melting, and melting is an endothermic process. The reverse process from liquid to solid we refer to as freezing, and this is an exothermic process. So remember, endo means within, so energy has to go in to the process. Exothermic means release of energy or release of heat. Okay, next, converting a liquid to a gas. So liquid water converting to gaseous water or water vapour, we can refer to that as evaporation or vaporization. And we've already seen that this is an endothermic process, so energy has to go in to that process, and the reverse is condensation, an exothermic process. And finally, converting a solid to a gas. So this is essentially skipping that liquid phase altogether. Directly from a solid, and this example is solid carbon dioxide, converted to gaseous carbon dioxide. This process is called sublimation. And everyone would be familiar with this, say at weddings or concerts. You'll have seen solid carbon dioxide. It doesn't turn to liquid in the container that's holding it. It will immediately undergo the sublimation process into gaseous carbon dioxide. The reverse process is called deposition, when gaseous carbon dioxide makes that transition into the solid CO2 phase. And you can see some dry ice pellets here, which represents solid CO2. But there's a natural process that occurs that involves deposition. This process where gaseous molecules form solid molecules. Can anyone guess what that is? And there's a clue here in this photo. Natural process of deposition. It occurs quite often in winter and spring.
Is it frost?
Frost, correct. The formation of frost is an example of deposition when that process occurs very quickly. All right, that completes our lectures for tonight. How's everyone feeling?
Yeah, not too bad.
Not bad?
Might have to, brush up a little bit, but
yes, yeah. And I'm feeling very tired. after all that. All right, so here are your tutorial questions based on this week's lectures. Don't know why it's skipped right down there. Okay, this set of questions is not assessed. There you go, tutorial 5. Not assessed, but make sure that you check your answers using that non-assessed online quiz. Remember, the non-assessed quizzes do remain open for the entire semester, but I strongly advise against students just waiting until the end of semester to do the non-assessed quizzes. That's really not the best way to revise and understand the material. So if you can keep up even with these non-assessed quizzes on a weekly basis, that will make your life much easier going into the final exam. Now, before you all go, I'd just like to point out something. All right, weekly prac students, when is your report number 1 due? Do we have any weekly prac students here? Oh yes, Joe. Joe, you're a weekly prac student, I believe. Do you know when your first prac report's due? Oh, there we go. Thank you, Joe. Very good. So 2 weeks from today at 9:15 a.m. Now, keep in mind, everyone, that the submission time should be observed very closely, because any late reports that come in will start attracting a penalty of 5% for every day or part thereof that it's late. All right, here we go. This is the section that I wanted to refer you to. So when you get up to gases, liquids and solids, these questions here, questions 1 through to 4, make sure that you use that formula PV equals nRT. You should be able to use that formula to solve all these problems. And remember what this means, STP. Okay, so we've covered prac report. Next week, does everyone know what next week is?
Easter Monday.
Easter.
Yes, it's Easter, and also next week is the mid-semester break.
Okay.
Okay, so there are no classes next week.
It's just the 1 week, isn't it?
Yes, just the 1 week. Just the 1 week, mid-semester break. And remember, that's a break from classes. It's not supposed to be a holiday.
Mm-hmm.
So it's a really good time to catch up with any work that you've fallen behind with. I'm sure it'll be a time when students are writing their first prac report. That is weekly prac students, not the weekend prac workshop students. So no online class next week, and there are no face-to-face classes. Okay. So make sure that no one turns up to Epping campus next week. Okay? Otherwise, you'll be very disappointed. Well, you might be happy that you get to go home, but if you're coming from a fair distance, you won't be happy.
Yep. Okay.
All right. So has anyone got any questions? Because I won't speak to you again now for 2 weeks, unless you send me an email. Yes.
The vapor pressure stuff kind of went over my head a little bit. Is there anything else I can read just to kind of...
Absolutely. Do you have any of the biology or chemistry textbooks?
No, I did borrow it from the library, so I might do that.
Okay, yes, yeah. Any biology or chemistry textbook, well, in particular, a chemistry textbook will explain vapor pressure, but you can also do a quick Google search and find an explanation of vapor pressure, but make sure you go to a reputable website. So, for example, if you find a website from any of the universities, I would trust the information given there.
Okay.
That's probably the quickest way to get the information, but definitely borrow one of those books again. They'll really come in handy.
Thank you. Just on that subject,
yes.
Once that if someone were to be unprotected in space, they would boil, and it never made sense to me. Why?
Sorry, can you repeat that?
If a person were to be stranded in space without a suit, they
Yes.
would boil. And
The
I never
per-
understood it because I always thought space was cold. But is this why they would boil? Because of the vapor?
So someone said that the person would boil.
Yes. Your blood would boil. That would be the instant reaction, and that's how you would actually die.
Ah, right. Okay. Yeah, look, that's not something that I'm familiar with. Yeah, I'm not sure about that. I would have to actually research that myself. But interesting that you asked the question, because one of the students this morning asked a similar question about boiling a cup of tea on the moon, for example. So interesting that you've come up with a similar or related question. Yeah. No, very good.
There was no collaboration.
No. No collaboration? No conspiring? No, good question though.
Silvana, on the
Yes, Liz.
tutorial from last week,
Yes.
specifically the biological molecules, I found that quite hard, and I got quite a few of the questions wrong in the quiz, even the second time around. Is it possible to get feedback on that, just so that I can...
Absolutely. If you would like to just send me an email and just tell me which questions you had problems with, and I can help you with that. Not a problem at all. This is what students tend to do. Unfortunately, not as many students as I would like to see, but students will send me an email and say, I really don't get question 19 and 21 or whatever. And I can usually clear up any confusion regarding those questions.
Perfect. Thank you. I'll do that.
Yeah. So please do that. Happy to help. Definitely. Any other questions for me?
No.
No? Okay. Everyone looking forward to the weekend prac workshop?
Yes, that's in 3 weeks or something.
That's in, I think 3 weeks, just under 3 weeks.
Okay.
Yes. Prac workshop. It'll be terrific.
Yep. Should be good.
Yeah.
Yes. With the company that delivers the lab coats to you, do you know if it's too late to order one through them for that weekend?
No, it's not too late.
Thank you.
No, you can order online anytime. Yeah, but make sure you don't leave it until the week before the actual prac workshop.
I think I'll do it now.
Yeah.
Okay.
Thank you, everyone. If there's no more questions, have a lovely Easter.
Yep, you too.
And a lovely mid-semester break. And I'll see you all in 2 weeks' time.
All right. No worries. Thank you.
All right. Thank you. Good night.
Good night. See you guys.